Vamos a dar paso a la siguiente ponente. Su nombre es ARACELI Aguilar González y va a presentar su trabajo titulado Optimization of a secretable murine GAA through lentiviral vectors, rescue Pompe disease cellular models. Cuando quieras, puedes empezar.
Muchas gracias. Well, I'm going to do the presentation in English. First of all, I would like to thank the organization for selecting me. I'm Araceli Aguilar, and I'm going to talk about the optimization of a secretable murine GA through lentiviral vectors, rescue Pompe disease cellular models. First of all, what is Pompe disease? This is a rare disorder caused by mutation in the lysosomal acid alpha-glucosidase gene. This enzyme breaks down the glycogen into glucose inside the lysosome. In this disease, the absence of this enzyme causes an accumulation of glycogen inside the lysosome, affecting cellular and muscular function, causing organ failure, affecting some tissues like the skeletal and the cardiac muscle, the central nervous system, and the liver. Depending on the age of the onset and the severity of the disease, there are several forms of this disease: the infantile or the classic one, the juvenile, and the adult. The most severe cases are the infantile and the juvenile because the percentage of GA activity is less than 1%. Nowadays, the actual treatment for this disease is the enzyme replacement therapy. This is approved by the FDA and EMA. It is based on an intravenous infusion of the recombinant protein of the human recombinant GA. However, there are several problems, like its cost is very expensive, and there are some severe side effects, like the immune response against the exogenous recombinant protein, and this recombinant protein don't cross the blood-brain barrier. The recombinant protein is captured by the mannose-6-phosphate receptor in the cells, and some companies are working doing some modification to increase the capture and the stability of the recombinant protein. Here I show you some of this kind of modification. However, they have some problems. So as an alternative, our group is working with gene therapy in this disease. We propose the hematopoietic stem cells as Trojan horses. In this case, we obtain this kind of cells from the patient. We modify the cells ex vivo with the transduction with lentiviral vectors, and we reinfuse these kind of cells modified into the patient, achieving cross-correction. With this modification, we obtain immunotolerance, we reach the central nervous system, stop the muscle weakness. To understand better the function of the GA protein, let's talk about the MGA maturation. The protein is synthesized in the reticulum endoplasmicum. Then it's transported to the Golgi when we have the precursor of 110 kilodalton. Then it is transported to the lysosome when it is processed to the most active forms, the 66, the 76, and the 60 kilodalton forms. This pathway, the lysosome, the pathway to the lysosome is around the 90% of the total protein. However, 10% of the protein that it is secreted outside of the cell, like the precursor form. And we have to take this into account because this protein that is secreted can be uptaken by the cells that are surrounding. So the main objective for this project is the designing of different chimeric murine MGA proteins to increase GA therapeutic activity in gene therapy strategies. To investigate the potential improvement of the design chimeras, we generate different knockout cell models in murine cells, skeletal muscle cells that are solate, and we study GA expression and secretion in different murine and human cell lines and the uptaking in the models that we have generated. First of all, we design different murine GA protein because as I have explained before, the percentage of the protein that is secreted it only the 10%, but we want to increase this percentage. For this reason, we designed 3 different chimeras protein containing the murine GA protein, the cDNA, that was codon optimized. The first construction also contained a leader peptide in order to improve the secretion to the media of the protein. But another construction contains the lysosomal signal peptide in order to target the protein to the lysosome. And our third construction contains a fragment of the EGF2 that is like a comparative construction to the actual treatment for the enzyme replacement therapy. With the 3 constructions, we make a 3D structure modeling of the proteins in order to know that our modification modified or not the protein. And the part green is the modification that we introduced, and we could see that our modification doesn't modify the structure of the protein, and didn't modify the active site too. So then with the 3 construction, we produce lentiviral vectors, and we want to transduce different cells. When we modify our cells, we are going to produce the protein, the GA protein, and this protein is going to be secreted to the media with the modification as we have introduced, and we want to improve the uptaking for the cells that are surrounded, then they are not modified in order to capture the protein. The second objective is the generation of murine muscle cell models, knockout cell. For this aim, we use solid cells that are skeletal muscle cells as a model. And for this aim, we look for the mutation most severe in Pompe patients. And these most severe mutations, we check that they are conserved in the mice sequence. With the CRISPR-Cas9 system, we design different guide RNAs for the different regions, and we want a knockout for the different mutations. For this aim, we nucleoffected our cells with the CRISPR-Cas9 system. We obtained the genomic from the cells, and we checked that our vision efficiency was high enough by the eyes and the T7. When we have our cells nucleoffected, we continue clonal analysis in order to obtain a population. Then, when we check the GA activity and we compare with the wild type, we see that there is a reduction. There are almost no activity in all the cases compared with the wild type. So we have generated the knockout models. Then we want to check if our 3 chimeric proteins were expressed, secreted, and uptaken. For this, we check the GA activity because we want to know if the protein is active or not. Let's focus first in the expression and secretion. First, for the expression, we transduce not only human cells, but also murine cells that are our final aim. We make some Western blot, and we could see both forms from the protein. The not processed one from 110 kilodalton, and the most active one about 76 kilodalton, or 70. In all the cases, when we check the GA protein, we could see that in all the cases, the lentiviral vector expressing the GA chimera containing the leader peptide, the IFG, is the most expressive one. What happened with the secretion? In the same kind of cells, when we plate our cells and we take the supernatant from these kind of cells and we precipitate it for the Western blot, we see the band of the precursor, which is the one that is secreted, And when we measure the GA activity, we have different results, depending on the kind of cells. In the human cells, the construction containing the S2G is the most secreted form due to its high homology to the human IGF2. However, in murine cells, the SIFG is the most secreted form in this kind of cells. But for our approach that we are interested in murine cells is that lentiviral vectors containing the expressing GAA chimera containing the leader peptide, SIFG, show improved secretion, as in expression. And finally, we want to check if our protein is uptaken by our cellular models knockout. For this aim, we plate our cells, K562, that are human cells, in our plate, very concentrated. We took the supernatant and we played in our cellular models. And after 24 hours, we measured the GA activity. In all the cases for the different clones that we have generated, we could see that the most secreted protein was more efficient rescuing GA activity in our models, because in K562 cells, the S2G were more secreted to the media. However, our result show that we have very low levels of recovery of the GA activity. And what could happen in this kind of cells? That there are no expression of the mannose 6-phosphate receptor in the cell membrane, like in Pompe patients. But we have to study more about that. So finally, the conclusion of this project is that we have generated different chimeric murine and GAA protein to investigate the better approaches to increase GAA therapeutic activity in gene therapy strategies. Using CRISPR-Cas9 technology targeting the MGA gene, we have generated different murine skeletal muscle cell lines, solids, that show absence of GAA activity. GAA expression and secretion were analyzed in human and murine cells, as well as in the knockout cells, and the uptake. And our results show that the GAA chimera harboring the leader peptide, called IFG, presented the best characteristics for its use in gene therapy approaches. And finally, I would like to thank to all my group and to the center and our collaborators. And if you have any question.
So thank you very much for your presentation. It has been very interesting. Do you prefer that we speak in English or Spanish?
I don't mind.
Okay, perfect. So I would like to ask you or if you have thought already in the next step of your work, if you know.
Yes, because this is the first step. Well, we are working some in vivo and in vitro. This is the first step in vitro. But we are going to translate this construction to transduce our cells, our hematopoietic stem cells from the mice, because we have a mice model of Pompe disease, and we are going to translate this experiment in vitro for the in vivo model.
Ok, perfect. So, vamos a dar paso a la siguiente ponencia. Muchísimas gracias.
A vosotros.